Hi, how's it going? My name is Jenna Gomez. It's at Stanford University School of Medicine, Department of Genetics, in the Snyder Lab. The presentation today is going over genome language modeling transforms medicine with representational power with regulatory genomics. I'll say that one more time just because it's kind of a mouthful. So it's genome language modeling transforms medicine with representational power for regulatory genomics. All right, so GLMs convert genome from raw nucleotide sequences into machine readable language that AI can learn. The genome language modeling overview shows that DNA speaks the language of life itself. So DNA sequences like GA, TTA, CA have tokenization, transformation, and machine learning models, as well as deep learning models. So DNA is treated as a language. Nucleotides converted into tokens analogous to words in natural language. Tokenization is a numerical representation that are transformed into vectors, features that language modeling, machine learning models can process. Okay. So the sequences are first tokenized using various tokenization methods. These tokens are converted numerical vectors for further processing. ML/DL models are numerical features are fed into machine learning models, and ML and DL models can learn genomic patterns. Representations can feed predictive models for sequence classifications, regression, and other genomic tasks. So there's downstream applications in predictive modeling, sequence generation, integration with other modalities, EHRs, and functional annotations. But the end goal is biological interpretation, so the GLMs can support functional annotations and predict genome features from sequence information. Right. The GLM pipeline progressively transforms biological sequence tokens, numerical features, learned biological information in a DNA sequence that is turned into chunks. So it's split into smaller chunks and then tokenized and k-merized into three-mers. And then you have their stop words removed, or certain stop words that are removed, or what were called stop codons. And then there's DNA cleaning. So DNA cleaning into from three-mers that are cleaned with the stop words. So all the stops codons are removed, and then you have your final k-mers. So that's pre-processing. So pre-processing in long genomic sequences are fragmented into manageable regions, and low information characteristics such as in can be filtered. And tokenization is DNA converted into a sequence, for example, overlapping three-mers, analogous to stop words or subwords. And the representation tokens can be encoded in traditional approaches such as um, ordinal or one-hot encoding or transformed using frequency-based embedding and neural network methods. Downstream modeling is the result of numerical representations, inputs for ML/DL models that are used for genomic prediction and annotation. So the traditional sequence encoding methods are tumors, and these are used for simplicity. So before we were challenged with the fact that we had to use three letters at a time, that three mers. We worked really hard to get six mers, 4, 5, and 6. But now we're learning that if you use two mers for simplicity, we can have AT, AT, TA, AT, TC, CG, 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 and GC and GA tumors that are used for simplicity. Ordinal encoding, you can see the encoding becomes a numerical representation for each ordinal encoding, and one-hot encoding, ATTA, ATTC, CG, GC, CG, and GA, in one-hot encoding becomes an array of numbers. And then for transformation of tokens, frequency-based representation, we have count vectorization, TF and TF-IDF. So those are frequency-based representation. An embedding-based representation, we have an embedding layer, word2vec, glove, and Elmo that can all be used as embedding-based representations. And lastly, we have neural network-based representation, transformers that are BERT and GPT, which are well-known transformers. And then tokenization is critical design choice because it determines what GLM considers biologically meaningful, like the word. So we have levels of tokenization, a DNA sequence, AAG, GT, CA, T, GT. So that's a DNA sequence right there. So you can see that the letters are levels of tokenization in a sequence. So the word or K-mer level tokenization has a 3-mer. And then there's also character level tokenization, subword tokenization, which we can see there. So it occurs at multiple scales. It can be represented by individual characters, K-mers, or larger subword like sequence units. All right, so subword methods address limitations of K-mers. They provide more flexible vocabularies and can better handle rare or previously unseen sequences. So BPE iteratively merges the most frequent neighboring sequence units to construct a compact vocabulary. Unigram word piece, sentence piece are alternative strategies that optimize tokens or tokenization that select using probability scoring, flexible word modeling. These approaches are increasingly relevant to genomic foundational models. So we have BPE that are unigrams, sentence piece, and word piece that all are types of subword tokenizers. Here are examples of different GLM algorithms. So algorithms in comparison, we have the aspects of the algorithms, the ordinal encoding, one-hot encoding, word embedding, and BERT, which are, I won't go over right now, but you can see them in the table. These are different comparisons of algorithms. And what you would want to do is encode them. All right, so frequency methods quantify how often a genomic pattern occurs, but not necessarily when it means in sequence context. All right, so transformation of tokens using frequency-based representation. We have 1 count vectorization, 2 TF IDF, which is an inverse document algorithm or transformation. And the TF score is the frequency of token, total number of tokens in the document. IDF score, the IDFs get scored through a log function where you have the total number of documents, frequency of documents containing a token. And then TF, IDF, and score. So the TF IDF is a matrix. The score is a matrix where you have a multiplier, a multiplication of the TF and IDF. All right, so count vectorization converts a DNA sequence into a numerical vector based on how frequently each token occurs. So TF, IDF weights and tokens according to both their frequency within a sequence and their relative uniqueness across sequences. These approaches provide interpretable numerical representations that can be used downstream in ML algorithms. The major limitation is frequency-based representation that largely captures a token token abundance rather than the semantic contextual relationships between genomic elements. So it's the frequency methods quantify how often a genomic pattern occurs, but not necessarily what it means in sequence context, and that's part of the frequency methods. And the word2vec moves a GLM beyond frequency counting to learn relationships between genomic sequence elements from context. So the word2vec model, a training sample is in a K-mer and a window size. And we also have word embeddings. And finally, a continuous bag of words and skip-gram model. So this continuous bag of words we'll go over in a little bit. The word-to-vect, this word-to-vect model, or word-to-vect learns genomic embeddings. These are the k-mers. They're able to embed the k-mers represented as dense vectors based on surrounding sequence context. So the context windows, the example of the windows is two neighboring k-mers that learn the relationships between sequence elements. The embedding space is k-mers with similar contextual usage from clusters, whereas outliers represent distinct sequence patterns. And this SIBO versus skipgram is the SIBO predicts a central k-mer from its context, whereas the skipgram it predicts surrounding k-mers from the central sequence element. So we have two of them. In this example, they both use ATA, and the SIBO predicts the current token or the surrounding sequence, and this skipgram predicts the surrounding sequence based on the current token. So this is really a technology evolution. So the technology evolution, like what is a GLM? A GLM is DNA language and AI. And how does the GLM work? It takes a sequence and it processes it into tokens, features, and machine learning. So what constitutes a genomic word? Characters, k-mers, and subwords. How do we numerically represent those words? Counts or TF-IDFs into frequency-based features. How does the model learn meaning? K-mer context, embeddings, semantic relationships. So the lesson here is genome language models transform DNA from a symbolic sequence into contextual representation that AI can use to learn genomic grammar, predict biological function, and integrate sequence information with other biological data. Thank you for your time. Wanted to acknowledge my lab in the Stanford Snyder lab. Here's us back in 2025. Thank you for listening. Thank you for your time.